Hey, hi everyone. This is Vishal Boyer. Today we will see antibiotics which kills microorganism by inhibition of their cell wall synthesis. Now, why cell wall is so much important? Let us see the structure. As you can see, we are observing the cell wall in gram positive cells as well as gram negative cells. In gram positive cells, as you can see, the cell wall is obviously made up of peptidoglycan is very thicker as compared to the gram negative cells. Now, why the cell wall is having so much importance? Now, many bacteria have this thick, rigid extracellular walls to protect them from osmotic lysis. Now, let us see what do you mean by osmotic lysis. Over here, we are observing a bacterial cell and we are taking an example of glucose sugar. Now, in this, obviously, a glucose sugar is very important for a bacteria for the energy purpose. It will try to accumulate more and more sugar inside the cytoplasm. And in this process, what is happening? The concentration of sugar, that is glucose over here, is more inside the cytoplasm than the outer environment. So this creates an osmotic pressure inside the cells. Obviously, the water molecule which are surrounding the cells, they will move due to this osmotic pressure created in the cytoplasm of bacterial cell. They will move from the lower concentration of sugar to the higher concentration of sugar. Now over here, the lower concentration is outside and the higher concentration is inside. What will happen? This water molecule will try to enter into the cell due to the osmotic pressure created, as you can see. So what will happen then? They will move inside the cytoplasm and they will continuously move inside the cytoplasm. So what will happen? So as they are entering into the cell, this will obviously the volume of the cell will increase and cell will appear like this. What will happen next? If they continue to move inside the cell due to the osmotic pressure created, obviously cell will accumulate more and more sugar molecules for its energy purpose. What will happen? A kind of situation like this may happen in which if a cell is accumulating more and more if water molecules are getting more and more inside the cytoplasm due to the higher concentration of sugar, it will increase the water volume is increased inside the cell, and after certain time it will become non-bearable, and the water molecule will create a pressure inside the cytoplasm that will be so huge that cell membrane will break like this, and cell membrane will break and cell water molecule will come out, and what will happen? Cell lysis, and this will create a very disastrous problem for the cell, that is cell death. So how does a cell wall prevent this osmotic lysis then? So let us see. As you can see, due to the higher concentration of solute present in the cytoplasm, water will move inside the cell as we are observing over here. This will obviously create a pressure against the cell membrane from inside. If a cell wall made up of peptidoglycan is surrounding that cell membrane, this will this will allow the cell to withstand the pressure created by the large water molecule inside the cytoplasm. So in this way, cell wall prevents the cell lysis and ultimately the death of the bacteria. So in this way, this is beneficial for the cell. Now let us review some steps in biosynthesis of cell wall. This will help in our understanding that how does this cell wall inhibiting antibiotics kills the microorganism at certain steps. Let us see that. So over here we are observing three parts. One is cytoplasmic site, then actual membrane site, and then the cell wall site, which is out of the cell membrane. Now see this carefully. There are two important precursor. One is N-acetylmuramic acid and another one is N-acetylglucosamine. N-acetylglucosamine, as you can see from here, will be phosphorylated and reduced into N-acetylmuramic acid. Then some amino acids like L-alanine, D-glutamic acid and mesodiaminopimelic acid will enter and they will add to the N-acetylmuramic acid and we will get this molecule. Then ultimately in the next step, 2-D-alanine are added further and we will get this molecule which is now having a pentapeptide. So N-acetylmuramic acid with a pentapeptide is formed. What will happen next? This is transferred into the membrane. Now in membrane, this is received by, this molecule is received by this lipid carrier called as, it is called as bactoprenol. Bactoprenol will take this molecule, as you can see from here, and another molecule of N-acetylglucosamine will come over here. It will bind over here and we will get a molecule of this N-acetylglucosamine bound with N-acetylmuramic acid with its pentapeptide. Then from the membrane, this is the molecule N-acetylglucosamine and N-acetylmuramic acid is transferred to the outer side of cell membrane. So this bactoprenol will then do what? It will transfer this out of the cell membrane like this. So as you can see, these molecules like this are transferred out of the cell. There are some enzymes like transglycosylase which will form a bond between these two, as you can see from here, and this will form a polymer of peptidoglycan. So in this way, one polymer of peptidoglycan is formed. Now to make it, and over here we can observing the second molecule of peptidoglycan. To make it having the strength that cell wall has, a transpeptidation reaction happens. And a transpeptidation reaction will happen over here, as you can see, between these two penultimate amino acid present in the pentapeptide. Now this has been catalyzed by an enzyme transpeptidase so that a bond is formed between these two polymers, and this will enable to have that strength in the cell wall, and it will unite the polymers of peptidoglycan into one thick peptidoglycan macromolecule, and this will form a cell wall with a very powerful strength. It will help the cell to cope up with the osmotic pressure created inside the cytoplasm. So in this way, cell wall is made, and a cell will obviously will do what? It will prevent itself from osmotic lysis. Now let us see antibiotics that inhibits peptidoglycan synthesis. And in this way, if cell wall is not there, what will happen? The cell will get exposed to osmotic lysis and cell will die. And this will help us to kill many pathogenic microorganisms. So let us see those antibiotics which inhibits cell wall synthesis, in particular peptidoglycan synthesis. First one is very important one and very famous, penicillin. The next one is cephalosporins. Next, cycloserine, vancomycin and bacitracin. Now over here we are taking very few examples, the very famous and foremost one. So in this way, these are the list of antibiotics which inhibit the peptidoglycan synthesis. So in our next video, we will see how does each of this inhibits peptidoglycan synthesis with their mode of action exactly, and will help us to kill pathogenic microorganism. So thank you very much. Bye bye.